All right, hello everybody. Tonight we're going to talk about the history of the experiments that were done to determine exactly what it is that DNA does, and that DNA was the hereditary material within a cell. So let's go ahead and get started. Okay, so a little bit of quick background information. We've already in our unit on heredity, we already talked about what chromosomes were. We saw in mitosis and meiosis, we saw how chromosomes are divided in between different cells. What is a chromosome really? They could see chromosomes. Scientists could see chromosomes under a microscope, just like you did in your onion mitosis lab. But what were they made of? What were they really? So, a few things you need to understand. Ooh, let me close up a few things to get them out of our way on our screen. There we go. Okay, so chromosomes are densely packed, double stranded DNA molecules with hundreds of millions of base pairs. The strands of DNA are wrapped around proteins called histones. So let's take just a moment to break down this diagram that we have here. So, up at the top, we've got a chromosome. You recognize that shape. If we were able to look really, really closely at that chromosome, you can see all these loops of purple. And if we look really, really closely at those loops of purple, you can see all these little purple things that are sticking out. Those little purple things that are sticking out, those are the histones that are made of protein. And if you look really closely, you can see woven in the purple are these strands of blue in the diagram. Those strands of blue are the DNA molecules. That's the DNA double helix. So you can understand why scientists were a little bit confused. They figured out that DNA might, or that the chromosome is the package of heredity, but what they weren't sure is whether it was through DNA or whether it was through proteins that our genes were being passed on. So we're going to look at a few experiments that kind of determined that information. Okay, so a little background information. By the middle of the 20th century, genetics was well established as a field of study. The problem, no one knew what genes were made of or how they work. In fact, when Mendel was studying his pea plant experiments and others, he had no idea what a chromosome was. And yet, this entire field of genetics was designed around something they didn't know what it was. So they started observing chromosomes during cell division, and they're like, okay, that makes sense that that would be the molecule of heredity. They were aware that the chromosomes were made of DNA and protein, but like I said, they didn't know whether they were protein or whether it was DNA that was passing on the hereditary information. And actually, most scientists believed that it was the protein that was passing on that heritable information. So there's a series of experiments, and we're going to talk about a handful of them. The first one is Griffiths transformation experiment. And in this context, you need to understand that the word transformation is a very technical term, and I have it written down at the bottom, and I strongly recommend that you actually write that definition down at the bottom. Don't get fooled when you see on a test question something about transforming or transformation and you just think, oh, they were changed. It's very, very technical term, and we're going to talk about what that is in just a second. So this experiment took place in 1928. Griffiths is trying to figure out kind of information about bacteria. And so he does some experiments with mice. In the first experiment, look at the diagram. In the first experiment, he injects mice with a pathogenic version of bacteria that we refer to as the S strain of bacteria. And they were deadly. And he injects it into the mouse, and the mousey dies. In the next experiment, he injects a different type of bacteria into the cell, a control group in this case, that is not pathogenic. And so the mousey is healthy, it's fine. In the third experiment, he takes the pathogenic bacteria from the first experiment and he heat kills them. And then he injects them into the mouse, and the mouse is perfectly healthy. Interesting. But then in the fourth experiment, this is where the word transformation comes from and how we get this concept. He mixes the dead bacteria with living harmless bacteria. So he's got the dead S strain and he's got the harmless R strain. He mixes them together and he injects that concoction into the mousey, and the mousey dies. What they discovered, took some time, was that the bacteria, the dead bacteria, they were releasing their DNA into the environment, and the living bacteria, the harmless bacteria, were able to suck up the DNA, and whatever DNA they got inside of them, they developed those characteristics. So when the harmless R bacteria sucked up the pathogenic DNA of the S bacteria, they became pathogenic to the mouse. So if we look at the definition down at the bottom, it says transformation refers to horizontal gene transfer. Horizontal means like from peer to peer, as opposed to vertical gene transfer, which is from parent to offspring. That's normal gene transfer. We expect that. But bacteria are actually capable of transferring DNA from one living individual to another living individual. I often give the analogy in class. Imagine if I got hit by a car and my guts were strewn all over the road. I'm sorry, that's really gross. But then one of my students came up with a spoon and started like eating my guts. And suddenly they had my characteristics. Well, that's in essence what these bacteria can do. They can suck up the DNA from the environment of a dead bacteria, and suddenly they are transformed. It's a very technical term. They are transformed by the new DNA that has been incorporated into their bodies. Now, that concept is going to come back again in the coolest way, because it turns out that's what allows us to do some really interesting genetic engineering experiments. So if bacteria can take DNA out of the environment, what happens if we take a gene for, say, human growth hormone, or a gene for human insulin, and we throw it into the environment where bacteria are? Sure enough, they suck up the DNA, and suddenly they are capable of making human growth hormone or human insulin. And we do it all the time. And we milk those bacteria then for insulin, and we package it and we sell it to diabetics. And we milk them for human growth hormone, and we package it and we sell it to people who need to use human growth hormone. Now, it's going to get really complicated. We'll learn about genetic engineering as we go through this unit, but that's just giving you sort of a foretaste of what's coming up. Okay, so then the next set of experiments is by Avery, McCarty, and McLeod. Do you need to know these names? No, they come up, and it's good to know these names as any kind of history, you needing to know names in history, but I'm not going to ever test you, and neither is the College Board on the names. So their experiment takes place in 1944, so we're about a decade and a half later. Their experiment, so it says here, they refined Griffiths experiment. So they're using the S and the R strain still. So if you take a look at this diagram, I don't need you to go into a ton of detail, but they heat kill the S cells, and then they treat the S cells with different enzymes that get rid of different parts of what's in the bacteria. So in this example, they treat the dead S cells with something called proteinase, and it destroys all the protein. So there's no protein whatsoever. And then they introduce the R cells, and the R cells are transformed. Remember, they don't know why the R cells are transformed. They're not sure what's going on, just that the R cells are transformed. So the R cells are transformed, and there's no protein present. So that means that protein is not the heritable trait, or the thing that's carrying inheritance. They add RNase, so there's no RNA available, and the R cells still transform into S cells. And then they add an enzyme that destroys the DNA, and now the R cells, they don't get transformed. And so this is pretty conclusive evidence that DNA is the trait of heredity. Is the trait of heredity? No, genetic material. That's a hard thing to say. Okay, so the DNA is the genetic material. All right, let's do one more experiment that really settled it for good. This is the Hershey Chase experiment. Again, these guys are so brilliant. Remember that they cannot see DNA at the molecular level. Nobody's got a microscope. Even today, we can't really see DNA at a molecular level. So what we're always doing is looking at phenotypes and then drawing conclusions and making creative experiments to see if we can solve what's happening at a molecular level. So their experiment is brilliant. They're going to use something that are called bacteriophages. Bacteriophages are viruses that attack bacteria. And on this diagram, they look kind of pink or grayish color. They look like little alien ships, and they are real, and there are infinity of them on our planet. I'm exaggerating. But they attack attack bacteria in the same way that the flu virus attacks us. They inject genetic material, and that's what scientists did not know what it was. Are they injecting protein or are they injecting DNA? We don't know. That's what we're going to figure out. But they inject genetic material into the bacteria, and that genetic material tells the bacteria how to build more viruses. That's all the genetic material says. And so the bacteria doesn't know any better. It just starts building more viruses because it's got genetic material that says build more viruses. So it builds and builds and builds and builds viruses until it explodes and the viruses come spilling out. That's the same thing that happens in your body when you get the flu. A flu virus enters into your cell. It gives your cell this genetic information that says build more flu viruses. Your body doesn't know any better, so your cell starts building flu viruses, and eventually your cell bursts open and it releases tens of thousands of flu viruses from just one infected cell. So, backing up, they take these bacteriophages that infect these poor little bacteria, and it says here they inject something into bacteria that tells the bacteria to build more phages. Whatever the phages were injecting, that was the molecule of heredity. So Hershey and Chase, this is their brilliant experiment. They decide to grow phages in two different, what we call media. In a medium is singular, media is plural. So they grow them in two different media. In the first batch, look at the diagram. In the first batch, they grow them in radioactive sulfur. Why? Sulfur is found in proteins. Maybe you remember from our biochemistry unit, disulfide bridges. One of the amino acids has sulfur in it. So proteins, their amino acids, some of their amino acids have sulfur. And so if you add radioactive sulfur, it's a way of causing proteins to kind of glow in the dark. So they have one batch of bacteriophages that are going to glow in the dark. Their proteins are going to glow in the dark. They grow another batch of bacteriophages in radioactive phosphorus. Why? Because DNA has phosphorus in it. If you remember the sugar phosphate backbone from our biochemistry unit, DNA has two strands and it goes sugar phosphate, sugar phosphate, sugar phosphate. So that phosphorus is now, they're making it radioactive so it glows. So any part of the DNA that is radioactive will be glowing. So then they allow the bacteriophages to infect bacteria. Don't ask what happens next. They literally blend it up in a blender. I'm not kidding. It's a blender. They blend it up, and then they put the remains in a centrifuge and they spin it. The material that's from the bacteria are the heaviest items, and so the bacteria are going to sink down to the bottom of the test tube. And what they discovered in the first batch, all the pellet at the bottom, that's the bacteria's contents. And there's no glowing, there's no radioactivity. And so that means there's no protein from the bacteriophages in those bacteria. There's nothing radioactive in the bacteria. In the second batch, they treat it the same way. They centrifuge it, and whatever's in the bacteria sinks to the bottom of the centrifuge tube, and it's radioactive. And that tells the scientists that what got injected inside of the bacteria was radioactive. So they have now conclusively proven that DNA is, in fact, the molecule of heredity. Okay, the next interesting experiment that takes place is happening in the 1950s. There's lots of others, but these are the classic ones. This is called Chargaffs Rule, and Chargaff starts studying all kinds of different organisms, because remember, all organisms use the same A's, T's, C's, and G's. And he starts looking at all of them, and what he discovers is that all species have different amounts of A's, T's, C's, and G's. So that's what makes us unique, is the order of our letters and how many letters we have. But the rule that he discovers is in every species, the amount of A is equal to the amount of T, and the amount of C is equal to the amount of G. Now, it's not perfect, but take a look here. Let's go with this one right here. So here's 17G and 17C. but over here it's 32.8A and 32.1T. So the amounts, the relative amounts of A's and T's and C's and G's are similar. This is what's going to lead the way to the discovery of the double helix shape of DNA, that the two strands were opposite, that A was always across from T and C was always across from G. All right, the next one we're going to talk about are Watson, Crick, Franklin, and Wilkins. So this is taking place in the 1950s, and they're going to get the Nobel Prize for this. These are competing teams that are rushing to figure out the actual structure of the DNA molecule. So Watson and Crick are using something called X-ray crystallography or X-ray diffraction data that was developed by Rosalind Franklin. Rosalind Franklin ends up dying of cancer and is not given the Nobel Prize. And we probably mentioned this earlier in the year. But they do figure out the double helix shape of the DNA molecule, and they receive the Nobel Prize for it in 1962. So now we've really figured out nucleotides on one strand are covalently bonded to each other, and that's called a phosphodiester bond. And I have that right here. You don't necessarily need to know the name phosphodiester, but it is possible that the word would show up on a test. And so I just want you to see it. So the hydrogen bonds, most of you remember when we did our biochemistry unit, that the left side of a DNA molecule is bonded to the right side with really weak hydrogen bonds. And that's going to become super important during this unit. And that's because the two sides can now separate, and we'll see that separation happen during replication when the DNA is being copied. So the hydrogen bonds that join the two sides together, they're very, very weak. The strong bonds are these bonds that are called phosphodiester bonds. So it's phosphodiester. The prefix di means two. So it's two bonds. You see my little lines pointing here and here. It's the bonds that occur between this is a sugar and this is a sugar, and then here's the phosphate group. So the phosphodiester bonds are the covalent bonds that hold the sugars together with a phosphate in between them. And those are covalent bonds. And so the two sides, I actually remember they're anti-parallel. The two sides are covalently bonded together, and they're super strongly bonded together. So those don't come apart. It's the opposite from each other that come apart. Okay, and then the last thing, just a quick reminder, again from our biochemistry unit, purines are the nucleotides that have double rings. So take a look at the diagram. So on the left-hand side are the double rings. Adenine and guanine are the purines, and the pyrimidines have a single ring, and these are the pyrimidines here. The way that I remember pyrimidines is that they have a Y in them. That sounds dumb, but the word pyrimidine has a Y, thymine has a Y, and cytosine has a Y. Unfortunately, uracil doesn't have a Y, so that's not that great of a rule. Thanks for nothing, KJ. Uracil replaces thymine in RNA, just to remind you. Okay, everybody, that's it for the history of DNA research. That we'll start with, let's see, DNA and DNA replication next. Have a good night.